All right, in this explainer, we are tracking an incredible mission in genetic engineering. And this isn't science fiction. We're following the real-world process of correcting a faulty gene in a living animal, starting from a single strand of DNA and going all the way to a potential cure. So, our story kicks off with this little guy, a dwarf mouse. Its growth is held back because of one single, defective gene that's supposed to make growth hormone. The plan is ambitious, to say the least. Take a healthy working copy of that gene from a human and introduce it into the mouse to see if we can get its growth back on track.
Okay, so the mission is clear, but how do you actually pull something like this off? Well, the strategy relies on a very specific type of gene therapy and an even more carefully chosen test subject. Now, it's really important to get this distinction right. We are not creating what's called a transgenic animal, where the genetic change is in every single cell and gets passed down to its kids. No, this experiment uses somatic gene therapy. That means it only targets specific body cells, in this case, muscle cells. And the key is, the change is not inherited. That makes the whole procedure a lot safer and more controlled for this kind of test.
You know, the choice of test subject here is absolutely critical. It's actually kind of a genius move. They used a special strain called a lit/SID mouse. That SID part stands for severe combined immunodeficiency. All that means is the mouse has a really weak immune system, it can't really fight off foreign invaders. And that's exactly what you want, because it means the mouse's body won't attack and reject the human gene we're trying to introduce, giving the therapy a real fighting chance to work.
Okay, so we've got our plan and we've got our subject. Now we need the most important piece of the puzzle, a clean, functional copy of the human growth hormone gene, or HGH. Let's break down how you find it and, more importantly, copy it. First things first, you have to get pure DNA from a human blood sample. Then, you bring in what are basically molecular scissors. These are special enzymes that cut DNA at very specific spots to snip out just the growth hormone gene. But one copy isn't nearly enough. So they fire up a process called PCR, which acts like a genetic photocopier on overdrive, turning that single gene into millions and millions of identical copies.
And this is the prize. This is the exact genetic sequence we're looking for, all 1,637 base pairs. It's the complete instruction manual for making human growth hormone. And it's super important that this includes everything, not just the coding parts, known as exons, but also the non-coding introns. Think of those as the director's notes in the script. They help control how the gene gets expressed later on inside the mouse's cells.
Okay, this right here, this is our first big mission accomplished moment. What you're looking at is a technique called gel electrophoresis. It sorts DNA by size. See that ladder on the left? That's our ruler. And that really bright white band? That's our gene. It's sitting right at the expected size, around 1,475 base pairs. This is the proof. The photocopier worked perfectly, and now we have millions of clean copies ready for the next step.
So we've got millions of copies of the gene. But you can't just, drop it into the mouse and hope for the best. It needs to be packaged in a way that the mouse's cells will actually accept and use. So the next step is to build a kind of biological delivery vehicle. And here's our delivery vehicle. This is called the PUC19 plasmid. It's this tiny circular piece of DNA that bacteria have. Seriously, think of it as a biological cargo drone. It's small, it's easy to work with, and, this is key, bacteria love to copy it in massive numbers. It's perfect for what we need. But a delivery drone needs instructions, right? The package also needs an on switch. That's what this is. A special sequence called the UBC promoter. Its only job is to basically scream at the cell's machinery, hey, read this gene, read it all the time. This makes sure the gene will always be active once it's inside the mouse.
So let's recap. We've got our three pieces, the plasmid drone, the promoter on switch, and the HGH gene message. To stick them all together, we use a process called ligation. If we used molecular scissors before, now we're using molecular glue. It's an enzyme called T4-DNA ligase, and it stitches all the pieces together into one perfect circle.
And voila! Here is the final product. The plasmid vehicle now contains the on switch and the growth hormone gene, all assembled in exactly the right order. This is our complete therapeutic package, now ready to be mass-produced.
And how do we know it all came together correctly? You guessed it, back to the gel. This confirms our success. Check out the lane on the right. The smaller, individual components are all gone. Instead, we have this one new, much larger band right at 5.1 kilobases, which is the exact combined size of our three parts. The molecular glue did its job.
Having one correctly assembled package is great, but for gene therapy, we need billions. To get that kind of scale, we turn to some of the smallest and most efficient factories on the planet, bacteria.
The whole process is called transformation, and it's pretty slick. First, E. coli bacteria are made competent, which just means their cell walls are made a little bit leaky. We mix these bacteria with our plasmids and then give them a heat shock. This quick blast of heat opens up tiny pores in the bacteria, and they just slurp the plasmids right up. Each bacterium that takes one up becomes a tiny factory, churning out our gene package.
So after the bacteria grow overnight on a plate, we come back to this. Each one of those dots is a colony that grew from a single bacterium. But wait a second, some are blue and some are white. The crucial question is, which ones are the right ones? Which colonies actually contain our complete gene package?
And the answer lies in this really clever trick called blue-white screening. See, the plasmid was designed so that our gene gets inserted right into the middle of another gene, called LAC-Z, which normally makes the colony turn blue. So if we inserted our gene correctly, it breaks the LAC-Z gene so no blue color is produced. That means the white colonies are the winners. They're the successful factories, the ones containing our complete therapeutic package.
We've now used our white colonies to grow vast quantities of bacteria, and from them, we purified billions of copies of our gene package. So now it is time for the final, most critical stage of the mission, delivering the cure to our patient.
The delivery happens in 3 steps. First, the mouse's leg muscle is pre-treated with an enzyme that makes the tissue more receptive. Second, our plasmid solution is injected directly into that muscle. It's a great target because the cells are really long-lasting. And finally, a technique called electroporation applies gentle electrical pulses. This briefly opens pores in the muscle cells, allowing all those plasmids to get inside.
So after all that meticulous work, all the cutting, pasting, copying, and injecting, did the mission succeed? Well, this one number tells a pretty powerful story. The treated mice saw a body weight increase of up to 33.1%.
And it wasn't just weight. The mice showed significant restoration of their skeletal growth, their overall size. The results were clear. Blood tests confirmed that the new gene was active, producing human growth hormone that circulated in their bodies for over 2 months after just a single treatment. The science, flat out, worked. This entire journey, from a genetic code on a computer screen to a real physical change in a living animal, it's just a powerful demonstration of what gene therapy can actually do. It shows a potential pathway for correcting genetic disorders right at their source, which leaves us with a really critical question. As this technology becomes more and more refined, what's next for human medicine?